Hello everyone, I welcome you all to Ganesh IAS Academy. In today's session, we'll be discussing science and technology based current affairs from April 1st to April 11th, 2024. Let us get into the details of today's content. The first news that we are going to discuss here is Mother of Dragons Comet. Why is this comet, that is Mother of Dragons Comet, is in news? And what is its significance? These details we must have to understand. Let us understand more details about Mother of Dragons Comet. This comet is officially named as Comet 12P or Pons-Brooks, and it is making a rare appearance in the northern hemisphere skies. That is the reason why it is in news. After so many years, it is making a rare appearance in northern hemisphere skies. That is the reason why it is in news. And what is a comet? We are talking about comet, but what is a comet? It is a cosmic body which is made mainly of ice, dust or rocky material that is traveling around the sun in an elongated oval orbit. So they are traveling around the sun and they are from distant origins, okay, and then it is mainly made of ice, dust and rocky material, okay. Moreover, this Mother of Dragons is a unique Halley type comet. So what is Halley type comet? We'll understand that in the upcoming slides. And this Mother of Dragons completes its orbit around the sun every 71 years and is visible after dusk, marking its first appearance since 1954. So in the year 1954, for the first time we saw this comet, okay, that is Mother of Dragons, and after that we are seeing it now after 71 years, okay. So this is what we need to understand from this particular topic. And what else? It falls under the classification of Jupiter family comet. Why is it falling under Jupiter family comet? That is because it is affected by the gravitational force of the Jupiter. That is the reason why it is called as Jupiter family comet. And then its nearest approach to Earth will occur in June 2024. In the month of June in 2024, this year, the nearest approach of the comet can be visible. And these are all the different sections or different parts of the comet. The structure of the comet is like this, okay. So you can find plasma tail, hydrogen envelope, nucleus of the comet, and then there will be dust tail. So there will be plasma tail and also dust tail, and there is coma, okay. So these are all the different parts of a comet, okay. A question based on this can also be asked. Next is about the Halley comet that we discussed about. We discussed that this Mother of Dragons comet is a Halley comet. So what is this Halley comet? Halley's comet, also known as 1P or Halley, is most well-known comet in the solar system, and it has a periodic term, okay. So it is a periodic comet. What does it mean? That it has an orbital period that is less than 200 years. So it is a periodic comet which has an orbital period of less than 200 years. There we saw for Mother of Dragons comet, the orbital period is 71 years. So obviously it is less than 200 years. That is the reason why it is classified under Halley's comet, okay. And this Halley's comet was named by European Space Agency, and then this comet exhibits a bright green color due to the emission of emerald colored light from diatomic carbon molecules, okay. Moreover, it is notable for its cryovolcanic nature. What is cryovolcanic nature? Cryo because of the icy content that is present, and it also has volcanic nature, which means it will burst out, okay. So the comet regularly erupts, creating stunning display of brightness, okay. So all these are the characteristics of the Halley comet that we are discussing here. And the 12P comet, that is the Mother of Dragons comet, is also a type of Halley's comet, okay. I hope it is clear. The topic that we are going to discuss now is Punnett Square. What is Punnett Square? Why is Punnett Square in news? Let us understand the details. Punnett Square is named after the British geneticist, that is Reginald Punnett. What is it? It is a grid. Punnett Square is a grid that is used to predict the possible genetic outcomes of offspring when two individuals with known genotypes are crossed. Two individuals are there, their genotypes are very well known. We'll understand what is genotype, phenotype and other details in upcoming slides. Two individuals are there, their genotype is very well known, and what are all the possible genetic outcomes of the offsprings of those two individuals. This can be found with the help of the Punnett Square. It is like a grid. We'll understand every other details of it. So here, what is genotype? Genotype of any organism is its complete set of genetic material. So the complete set of the genetic material of a particular organism is genotype. Moreover, genotype can also be used to refer to the alleles or variants of an individual carries in a particular gene or genetic location. Okay, so even alleles and variants an individual carry in a particular gene or genetic location is also called as genotype. Okay, and it consists of a simple grid. What consists of a simple grid? This Punnett Square consists of a simple grid with squares that is representing possible combination of traits that is inherited from each parent. Okay, each parent will be contributing to different traits of the offspring or the child. Okay, so what are all the possible combinations that can be there? This is got with the help of Punnett Square. Okay, what else do we have to understand? We need to get into the basics of the Punnett Square. First, we need to understand the concept of allele. We saw the term allele, and then we saw the term genotype, and then we must also understand the concept of phenotype and how is phenotype and genotype different. Okay, so first we'll understand what are alleles. Alleles are nothing but one of two or more alternative forms of gene that arise by mutation and are found at the same place on the chromosome. Okay, so they are alternative forms of the same gene that arise by mutation and they are found at the same location in the chromosome. So this is the chromosome and I have one allele here and another allele here. Okay, all these are alleles. So what is an allele? Allele for white flower is expressed by this color. Okay, and allele for purple flower is expressed by this color. Same gene, same location, but different variants of the same gene. Okay, same chromosome are there, and then this particular gene alone is varying. This variation in the gene is called as allele. Okay, so the locus of flower color gene is here. So this is where the gene which is responsible for flower's color will be present. That is the location. Okay, here we have for white flower and there you have for purple flower. So this is how it is varying. So this is allele. Okay, and what is phenotype? Phenotype is nothing but the set of observable characteristics of an individual that is resulting from the interaction of the genotype that we discussed with the environment. So what is genotype? Genotype is an organism's genetic information, as simple as that. Okay, an organism's genetic information is genotype. When that genetic information interacts with its environment, then that becomes phenotype because it will express itself in the form of observable characteristic. Okay, so observable traits, the physical traits are called as phenotype. Say for example, BB, that is homozygous dominant gene is genotype here. Okay, whereas it is expressed in the form of purple flower because purple is the dominant one here. And then capital B, small b, this is heterozygous. They are not same, so heterozygous. They are same here, dominant and dominant. That is why it is homozygous dominant. This is heterozygous. One small b and another capital B, that is dominant and recessive. Both are present here. Okay, it will express itself as purple again. This is the observable characteristic that we are talking about. That is phenotype. And when you see this homozygous recessive, Homozygous because both are recessive, B, small b and small b. White is expressed because small b is for white flowers. So this is phenotype because the observable trait that we are seeing, the physical trait that we are seeing. So I hope it is clear what is genotype, what is phenotype, and what is allele. Okay. Next, we'll summarize this. So genes determine traits. We know that our genes determine our traits or characteristics like eye, skin, hair color of any organisms. And each gene in an individual consists of two alleles. One comes from mother and another from the father. So which is going to be the dominant and which is going to be the recessive one? And based on the combination, you will be, your eye color or your skin color, hair color will be determined. Okay, this is the basic. Next is, some alleles are dominant, like we discussed, meaning they ultimately determine the expression of the trait. The dominant one will ultimately determine what trait will be exhibited. And then other alleles are recessive and are much less likely to be expressed. Okay, so if capital B is dominant and small b is recessive, if I have this combination BB, obviously this will be dominant. And even if I have this combination B and small b, capital B and small b, even here the dominant one will be expressed. Only when I have this combination, that is BB, both recessive, my recessive trait will be expressed. Okay, this is what we need to understand. Then when the dominant allele is paired with the recessive allele, that is what we discussed, the dominant allele determine the characteristic. That is the second case. When the dominant allele is paired with the recessive allele, the dominant one will be expressed. And when these traits or characteristics are visibly expressed, then they are known as phenotypes, that we discussed in the previous slide. And the genetic code behind these traits is known as genotype. As simple as that. Okay, I hope it is clear. Now let us understand what is Punnett square and how does it work and how can we construct a Punnett square. Okay. So the grid, how is the grid formed? First is we need to set up something. What is it? List the possible genetic traits of one parent along the top of the grid and the other parent along the side. So here I am giving one parent's trait and here I am giving another parent's trait. Similarly, this is another Punnett square. Here one parent's and here another parent's. The traits are given like this, and then combination has to be done. That is, fill in the squares by pairing each trait from one parent with the corresponding trait from the other parent. So this is how I will combine this. P, P, capital P, small p, capital P, small p, capital P, small p. So in this combination, if you see, 100% it will be purple because there is one dominant allele in every other combination. So if there is at least one dominant allele in a combination, that dominant trait will be expressed. Okay, but if you see here, it is capital P, small p, capital P, small p. This type of combination. Here if you see, capital P, capital P, capital P, small p, capital P, small p, and small p is also there. 2 recessive is also there. So here if you see, it is 3:1 combination. So 3 dominant trait and 1 recessive trait will be expressed. So these are all the different combinations. This is how we'll know which trait will be expressed or which type of phenotype will be expressed. Okay. Next is outcome. So each square represents a potential combination of trait that the offspring could inherit from the parents. So all these are just possibilities. So Punnett square is all about possibilities. These are all the different possibilities that can be expressed. These are all the different traits that can be expressed. Okay, I hope it is clear. Now let us understand the application of Punnett square. So Punnett squares are commonly used in biology to understand inheritance pattern, like we saw, like when you learn about the dominant and the recessive genes in school level. Next is, it is useful tool to help predict the variations and probabilities resulting from crossbreeding. So when I go for crossbreeding, all the different variations and combinations can be predicted. And then it can also be used to understand the genetic traits of the offspring in animals, okay, including humans. And researchers typically use them together with Mendelian inheritance method. Okay, so Mendelian inheritance method along with Punnett square. will give you excellent results, outcomes. Okay, I hope it is clear. The topic that we are going to discuss now is Agni-Prime Missile. Okay, why is Agni-Prime Missile in news? Let us understand the details. The Strategic Forces Command, SFC, along with DRDO, that is Defense Research and Development Organization, has successfully tested the new generation ballistic missile, that is Agni-Prime. From where have they tested this? From Dr. APJ Abdul Kalam Island off the coast of Odisha. Okay, Agni-Prime, and it is a ballistic missile. What are all the different features of this Agni-Prime, and how can we compare this Agni-Prime with previous missiles of Agni series? Okay, this is what we are going to know here from exam perspective. So it is a medium range nuclear capable surface to surface ballistic missile. Every other detail is important. Okay, so it is medium range. So what is the range here? So if you see, its range is from 1000 to 2000 kilometers. Okay, medium range. Next is, it is nuclear capable. So it can carry both conventional and nuclear warheads. Next is surface to surface. What type of missile is this? It is surface to surface missile, and it is also a ballistic missile. Okay, so you can differentiate it from cruise missiles here. Either it is cruise missile or ballistic missile. Here it is a ballistic missile. That is, it is going to take a ballistic trajectory. And then it was tested for the first time in 2021 June. That is Agni-Prime, and it is developed by DRDO, and we know the range of this Agni-Prime. What else do we have to understand? So this is Agni-Prime, and this is the Abdul Kalam Island that we are talking about. Previously, it was called as Wheeler Island off the coast of Odisha in Bay of Bengal. Next is, the missile is two-stage. All these details are also important. It is two-stage missile, and then it is canisterized missile, and then solid propellant ballistic missile. So they are using solid propellant. This is one information, and it is canisterized, and then it is two-stage. So here they are saying that it is canisterized. What is canisterization? Canisterization of missile reduces the time required to launch the missile while improving storage and ease of handling. Canisterization is nothing but placing the missile inside a tube kind of structure. So if you see here, this is a tube kind of structure they are talking about. This is canister here. And inside the canister, they will place the missile, and from the canister, it will be easily launched. So what is the advantage of using canister? That it reduces the time required to launch the missile, because if I have to launch the missile, I must have to place it in a perfect place, in a perfect location. Whereas if I place it inside the canister, I can take it wherever required. So this is there, and then storage and ease of handling is also there. When it is canisterized, when it is safely placed inside the tube, then it is safe. This is also there. And then even if one functions, I mean, if you see the second point, that is, it has this dual redundant navigation and guidance system. Why are they going for this dual redundant navigation and guidance system? That is because even if one functionality suffers a failure, then a backup feature would still enable the objective that is to be achieved. So it has a backup system. That is why they are being called as dual redundant navigation and guidance system. And then it is lighter than all earlier Agni series of missile. Agni-Prime is going to replace even Prithvi. Prithvi will be replaced by Agni-Prime, and Agni 1 and 2 will be replaced by Agni-Prime. Okay, so that is because it is lighter than other vessels. Next is, so we need to understand the basics of Agni missiles here. About Agni missiles, they are long range ballistic missiles that are designed for surface to surface attacks, and they are capable of nuclear payloads, which is very clear, we know that. And then what was the first variant of Agni series? The first missile in the series was Agni 1, which was developed under Integrated Guided Missile Development Program, which was developed by Dr. APJ Abdul Kalam, and it was tested in 1989 itself. And then this integrated guided missile program also has other missiles. What are they? Prithvi, Agni, Trishul, Nag and Akash. Patan or Patna, however you wish to remember it. Prithvi, Agni, Trishul, Nag and Akash. Patan or Patna. So these are all the missiles that come under integrated guided missile development program. And then this table is very important. Different Agni missiles, its type and its range. So first is Agni 1. It is medium range ballistic missile. See the range. It is 700 to 1200 kilometer and it is operational, which is they are being used now. And then comes Agni-Prime. Medium range, but it is under development because now only we are testing it at different ranges. Okay, so 1000 to 2000 kilometers. And then Agni 2, again medium range, 3000 to 3500 kilometer. Agni 3, this is intermediate range. This is 3000 to 5000. And then Agni 4, intermediate range, 3500 to 4000. And Agni 5 is intercontinental ballistic missile. This is intermediate, but this is intercontinental, which means its range is from continent to continent. See the range here, 7000 to 8000 kilometer, and it is operational, being used by India. And Agni 6, it is under development and its range is 11,000 to 12,000 kilometer. Okay, which is more or less equal to the diameter of the earth. Okay. And then this Agni-Prime or Agni-P is expected to replace Agni 1, Agni 2 and even Prithvi, because Prithvi is also surface to surface. The list that we saw in the previous slide, that is Patan, Prithvi, Agni, Trishul, Nag and Akash. Of these, Prithvi and Agni are surface to surface missiles, which is to be understood. The other two are surface to air, and then Nag is anti-tank missile. All these details are very important. Okay. With this, we are coming to end of this particular topic. The topic that we are going to discuss now is NEX CAR 19. What is this NEX CAR 19? It is India's first indigenous CAR T-cell therapy. So what is this CAR T-cell therapy? What is T-cell and what kind of cell is it? All these details we must have to understand. Let us get into the details. So President of India recently launched India's first indigenous anti-cancer CAR T-cell therapy, that is NEX CAR 19. Where was it launched? It was launched at IIT Bombay. Okay. So what is this CAR T-cell therapy? It stands for chimeric antigen receptor. Chimeric antigen receptor, that is CAR T-cell therapy. Okay, and then T-cell is being employed here. And then here it is clear that it is anti-cancer therapy. So it is a type of cancer immunotherapy that uses patient's own T-cells. So the patient's own T-cells are used. They are genetically modified. Those T-cells are genetically modified in the laboratory setup. Why? To enhance their ability to locate and destroy cancer cells. So locating and destroying cancer cells is my objective here, because our immune system at times will not locate and destroy the cancer cells, thus leading to proliferation of cancer cells. But this will not be, this will be hindered by CAR T-cell therapy. Okay. Now let us understand how does the therapy works, the working. So here T cells are white blood cells. They are a type of white blood cells that are responsible for identifying and fighting illness and infection. So the responsibility of T cell is to identify and fight any illness or infection. And each T cell has a receptor in it which can recognize antigens. Okay, that is protein or molecules that is recognized by the immune system, that is called as antigen, and they will be having a receptor. And here we are going to modify this receptor, that is what is chimeric antigen receptor, CAR. Okay, so cancer cells may have antigens that immune system does not recognize to be abnormal, allowing cancer to evade the immune response. Sometimes this might happen. So our immune system necessarily may not affect certain type of cancer cells because they cannot recognize the receptor. Okay, so they will not be having the receptor. So CAR cells are genetically engineered in the lab to express a new receptor that can bind to cancer cells and effectively kill them. That is the reason why it is becoming famous, CAR therapy. So what is the process involved in it? First is we need to collect T cells, how the blood is drawn from the patient's arm and then the T cells are separated from the blood using apheresis machine. Okay, and then engineering T cells. I'm collecting T cells and then I have to engineer or modify the T cells. How am I going to do it? So in a laboratory, these T cells are modified by adding manufactured chimeric antigen receptor, CAR, and then they are allowed to multiply and grow. After enough CAR cells are prepared, they are injected back into the patient's arm, that is infusing CAR T cells. And in this process, even chemotherapy can be integrated. So chemotherapy may be recommended before CAR cell therapy to enhance the treatment's effectiveness. And then this process can take place in outpatient infusion center or in the hospital setting itself. Anything can be done. This is the CAR T cell therapy process. So this is how it is done. Blood sample is collected and then it is reprogrammed. Why? To insert these receptors. These are all the receptors that has been introduced to the CAR cells, to the T cells, and then they are multiplied. After multiplication, they are infused back into the person, and then here it will effectively attack cancer cells. So this is CAR cell therapy. So this CAR cell therapy is effective against certain types of cancer, especially when other treatments are ineffective, and it is currently FDA approved for treating hematological malignancies, that is leukemia, lymphoma and multiple myeloma. Okay, hematological means one which is related to the blood related cancers, that is leukemia, white blood cancer, lymphoma, lymph cancer, and then multiple myeloma. All these can be addressed with the help of CAR cell therapy, and this next CAR that India has indigenously developed is designed to target cancer cells carrying CD19 protein. Okay, there are cancer cells which carry this protein, and in these cases this next CAR 19 will be most effective. And then this CD19 is actually a marker on the cancer cells, and this technique will enhance the precision in treatment, and it has been developed jointly by IIT Bombay and Tata Memorial Center. All these details are very important. CAR cell therapy, I hope it is clear. And then what is its effectiveness? So approximately 70% of the patients respond to this next CAR 19 treatment, with some achieving even complete remission from the cancer. Okay, and then lab and animal studies indicate low drug related toxicities. Okay, so there is low drug related toxicities when it comes to this therapy. Okay, and the trials for pediatric patients at present only for people above 15 age this is possible, but the trials for pediatric patients is underway at Tata Memorial Hospital, ensuring broader applicability in future. Okay, all these details are important from exam point of view, where you can use these in your answer writing also. The topic that we are going to discuss now is India achieves target to eliminate Kala Azar. So why is it in news? That is because according to the data from National Center for Vector-Borne Disease Control Program, India has successfully met its goal of eradicating visceral leishmaniasis. What is visceral leishmaniasis? It is commonly referred to as Kala Azar. So what is Kala Azar? How is it spread from one person to another? What is the vector here? All these details we must have to understand. So first, talking about this center which has provided the data, that is National Vector-Borne Disease Control Program. It was started in the year 2003-4. Why? To prevent and control disease like Japanese encephalitis, dengue, Kala Azar, lymphatic filariasis, malaria and chikungunya. All of these are vector-borne diseases. Okay, so National Vector-Borne Disease Control Program, they are coming under this. And then, despite persistent challenges, we face many challenges. Despite all this, India met its elimination goal initially set for 2010, but it was later extended until 2023. Only in 2023 we were able to achieve this target. This Kala Azar has long been a health concern in many states of India like Bihar, Jharkhand, West Bengal and Uttar Pradesh. Okay, and in the year 2003, India reported only 520 cases of Kala Azar, thus meeting the elimination criteria that was defined by WHO, that is World Health Organization. That is the reason why we are saying that we have met elimination target that is set by World Health Organization. Okay, we have effectively eliminated Kala Azar. Now let us understand the details of the science behind how does it work, what is the vector and what is the transmission, all these details. So Kala Azar, also known as visceral leishmaniasis, is a parasitic disease that is caused by Leishmania parasite. Okay, in India it is Leishmania donovani. Okay, and here it is a neglected tropical disease. India is a tropical country and it is a neglected tropical disease. That is, it is not given as much importance as many other diseases like malaria, dengue. That is the reason why it is called as neglected tropical disease. They are not given much importance for their control. Okay, and then it is the second largest parasitic killer in the world after malaria. So after malaria, Kala Azar kills many people. Okay, it is a parasitic disease, and then it is transmitted through the bite of the infected sandflies. So what are the vectors here? Vectors here are the sandflies. And what are all the symptoms of this disease? Fever, weight loss, enlargement of spleen, liver and anemia. And Kala Azar primarily affects spleen, liver and bone marrow. So what are all the different organs that are affected here? Spleen, liver, bone marrow. And what is the fatality rate? 95% if left untreated. So treatment has to be given at proper time. If left untreated, 95% fatality rate is there. That is, they are bound to die. And what is the treatment option that we have? First is injectable liposomal amphotericin B. Amphotericin B is the medicine that we have, injectable material that is. Next is 12-week course of oral miltefosine. What is this miltefosine? Miltefosine is prescribed for PKDL. What is PKDL? Post Kala Azar dermal leishmaniasis. So even after taking treatment for this leishmaniasis, we will be getting certain other problems as side effects. Okay, to address that, we are administering this oral medicine, that is oral miltefosine. Okay, so post Kala Azar dermal leishmaniasis, what is it? It occurs when Leishmania donovani invades and multiplies in the skin cells. So it is dermal. Dermal means skin, and it is causing skin lesions. PKDL typically appears after Kala Azar treatment. After taking treatment only we get affected by PKDL, and for that, for addressing it, we have this medicine. Okay, all these are important details to be noted. Next is, what are all the strategies that has been adopted by India to control or eliminate Kala Azar? First is spraying, intensive indoor residual spraying to target and reduce sandfly breeding grounds. So we have to reduce the sandflies breeding ground, and then wall plastering was also done. So coating walls with a specific soil called as Gerard soil to deter the sandflies from nesting. So their nesting and breeding has to be controlled. And then we also have treatment compliance protocol, okay, which was done with the help of ASHA workers. So engaging these ASHA network, that is accredited social health activist network, to oversee the treatment completion for PKDL patients, that is post Kala Azar, dermal leishmaniasis. Next, there are three different forms of Kala Azar, three main forms of Kala Azar. What are they? One is visceral leishmaniasis. Visceral means they impacting multiple internal organs and represent the most severe illness. So visceral leishmaniasis is the most severe illness. Visceral generally means the internal organs that we have, okay, inside the body. So multiple organs will get affected, will be impacted. And then you have cutaneous leishmaniasis, and it is the most common form, causing skin ulcers mainly on exposed body parts, which can result in lifelong scars and disability or even stigma. Lifelong scars will be there. Next is mucocutaneous leishmaniasis. Mucus will be lost. Okay, so it destroys mucous membranes in nose, mouth, and throat, thus leading to severe damage. So these are all the different types of leishmaniasis that we have. Okay, I hope it is clear. So with this, we are coming to end of today's session. I hope you found the session to be very useful and informative. Let us see in the next session. Thank you all.